All right, well, I think we'll get started here. Welcome everyone to another installment of our Sustainable Energy Seminar Series. If this is your first time, my name is Scott Williams. I'm the Research and Education Coordinator at the Wisconsin Energy Institute. My role is working in energy education at the Wisconsin Energy Institute. And the Sustainable Energy Seminar is something that we do every 2 weeks to bring together lots of different disciplines on campus, from science and engineering to social sciences and humanities, to talk about the many challenges in creating a more sustainable energy future and some of the solutions that folks are working on. And with the goal is really to provide a more holistic picture of how different disciplines understand energy challenges and develop solutions. This session is being recorded, so it will be available if you sign up for our bi-weekly email list. We'll send you a link afterwards to the video. This is also for the Certificate in Engineering for Energy Sustainability undergraduate program, as well as the Master's in Sustainable Systems Engineering. We'll allow, if you would like to ask questions of our presenter today, you can do so in the Q&A box, and I'll do my best to monitor the Q&A box during the talk and stop and ask the question if it seems like an appropriate time to interrupt. Otherwise, yeah, feel free to type in the chat, let us know where you're from, and introduce our speaker now. So today's presenter is Dr. Caryn Wadler. She is a postdoctoral researcher in the Thomas Lab, working in bacteriology, and part of the Great Lakes Bioenergy Research Center, which is part of the Wisconsin Energy Institute, which I'm at right now. And she'll be talking about what ways that we can take some of the waste products from biofuel production and seeing if we can use other microbes to develop some valuable products from that. So welcome.
Hi, Scott. Thanks for inviting me to be here. I'm really excited to talk to students, and there is no bad time to interrupt. Please don't let me talk over something important that you guys need to know to understand. So, as I was introduced, I'm a researcher in the Thomas Lab, and I've actually been working funded for the Department of Energy on biofuels research, actually through my PhD work as well. So I've been doing this for some long time, which has given me some extra insights into a little bit of what's going on here. So I'm just going to jump right in. And the first thing that I want to talk about is what is the GLBRC? So the Great Lakes Bioenergy Research Center is a collaboration of over 400 different scientists from a bunch of different universities. We have people over in Michigan, in Texas, we have people in British Columbia, and I think someone in Yale now because someone moved out there. And all of these people are working together to make biofuels and products out of plants more sustainable. And as you know, most of these researchers are here in Madison. There are 3 other big bioenergy research centers, all funded by the Department of Energy. There's CABI, which is down in Urbana-Champaign, Illinois, J-Bay, which is out near Berkeley on the West Coast, and the Center for Bioenergy and Innovation is linked up with Oak Ridge National Lab in Tennessee. And each of these research centers is going kind of towards the same goal. So we do a lot of cross collaboration, not just within the GLBRC, but within these different institutes as well. We have meetings with them and we share work and science. The mission of the GLBRC sounds deceptively simple at first, but the work is to develop economically viable and environmentally sustainable biofuels and bioproducts made from dedicated energy crops grown on marginal lands. And this is actually kind of one of the unique things about the GLBRC that I really love. They're concerned not just with getting a good biofuel, but everything that goes into making biofuels. So you'll have groups in the center working on everything from what lands are we going to use to grow the plants? How can we grow and engineer the plants so that they can be broken down more easily into products that bacteria can use to make biofuels or other microorganisms can use to make biofuels? And then how can we deal with the waste products from that? What biofuels are the most cost effective to make? And so on and so forth, answering all of these questions, but in a really collaborative way. So each group is kind of working with another group to make this whole pipeline be cost effective. And that's one of the things that when I first started working at the DOE funded research that I was really kind of surprised at. So when I joined, the DOE was doing this thing where they were funding a whole bunch of different research products, with the goal being just to see which technologies looked more promising so they knew what technologies to fund. Because for them, this was this new area of biofuels. How do we want to go about deciding what's the best thing to do? Let's put a bunch of money at it. Let scientists go for a few years, come up with the best technologies they can, and then we'll pick and choose. And when I went to a conference about that in, I say 10 years ago, I was really surprised that we had someone engineering a plant that was going to be able to be broken down by a different organism than they were trying to use at the time to make the biofuels. And the biofuel that they were making, they were engineering an engine to use a different biofuel because they were trying to optimize the engines as well to run off these biofuels. And just nothing seemed to kind of go together. It was just this whole, well, we have this product, we're going to try to do this, we're going to try to do that. And now when I go to one of these conferences, it's this whole pipeline and everything is working together and everyone is kind of considering, hey, can you guys engineer this? That'd be really great. And they're working ahead on based on what the other groups are going to do, which leads to everything kind of moving along a lot faster as well. So why did Wisconsin want to get in on this thing? Well, for Wisconsin, there's a whole bunch of really important motivators. So this will support Wisconsin's economy, farmers, manufacturing. Having a fully scaled biofuels industry provides a whole bunch of jobs. It provides support to rural economies, especially agriculture, because there's a lot of infrastructure that has to go into moving these sorts of feedstocks around once they develop where to put them. We could get energy independence, very important in the current days. And then just environmental concerns. If we can work on biofuels, then we can enhance soil and water health, reduce carbon emissions, and mitigate climate change, which is, of course, really high, lofty goals. So what I'm showing you here is the Kellogg Biological Research Station. This is where the GLBRC has been doing a lot of their work on the plants and the soil systems, which I think is really cool. And one of the nice things to see, I went to Champaign-Urbana myself, so having crops growing in the middle of school is something I guess I get used to. And so these are some of the bioenergy crops that they're working on. So right now, scientists at the GLBRC are studying whether to be working with switchgrass, poplar, or some other different things there. But ideally, they're looking for a native perennial, low input plant. So it's something that's going to be hardy, be able to be grown year after year without much maintenance, and then be able to be used for a feedstock for these bioenergy things. So I've talked a lot about this pipeline. What does this look like? So what this looks like is we have these marginal lands, which are marginal lands just mean lands that are really of no use for anything else. They're not protected habitats for any sort of creatures. They're not useful for growing crops. They're the sort of lands that if you were going to take them and do something else with them, it wouldn't really affect the environment very much and could be useful for us. You grow your energy crop, and then you have to break down that energy crop into something that microbes can ferment, which are these kind of sugars here, and then they use that to make biofuels. So I just wanted to talk to you a little bit more about how that process works, just to give you a little more background there. So when I talk about breaking down these energy crops, I just want to point out that plants are really, really hard to break down. We've all eaten plants. We know what it's like when you have to chew them. So in order for the bacteria and other microbes to be able to utilize these to make biofuels, we need to break down the plant cells, which are comprised of lignin, hemicellulose, and cellulose to get at the cellulose, which is shown here with these yellow fibrils. And that's the thing that the bacteria are going to eat. The lignin, not at all really, and the hemicellulose, it depends on what microbe you use. But each of these cellulose chains is actually made up of a bunch of glucoses, and glucose is the building sugar that all microbes enjoy eating. So what does this look like in our processing plant? Well, in our processing plant, we have to use this pre-treatment to break up the lignin and the hemicellulose to make that cellulose accessible to the microbes. So the first step of this is actually about what you think. We grind it up just in a big giant grinder. I've used ones that we have built into the wall. It's literally like a big giant grinder that you use to grind these up. And then you use heat, acid, pressure, salts, everything you can to kind of break up this lignin so that you can get at the sugars inside. And then we use an enzyme step. So this is actually a proprietary thing. We have to sign an NDA before we're allowed to work with anything that has touched the enzymes that they have used, because it's a very proprietary mix that they have developed specifically for this. And they use these enzymes to break down cellulose into pure glucose so that the microbes can use them to make biofuels. So that's this breakdown step of the process. And then the next step is fermentation. And this happens just in these giant batch reactors. We put our microbes in, we put in this treated biomass, and then we let them go. And out of that, we harvest biofuels. And we're also working on trying to get out of this bioproducts. And that's a lot of what I'm going to be talking about for the rest of the science portion of things today. Does anyone have any questions about how that initial part of the pipeline works? Are we good? No questions. All right. So you can use plant
We have one question
biomass.
here.
Yeah, go ahead.
What conditions do you use your enzymes under?
The enzymes are used, you mean just like, do we put them in the fermenter with our microbes?
I'm not sure. This is just what was typed into the Q&A box.
So we take the pulpy mash of the plant matter plus water to let them go. And then because there were salts and other things in there and temperature, which are all things you need to control for enzyme activity, we just inject those in there and let them go for a while to break it down. So we give them some time and then we add the microbes.
Okay, yeah, the follow-up question is what temperature and pressure can they withstand? Are impurities okay?
Impurities are okay. So they'll recognize hemicelluloses and cellulose and be breaking those down. I think they use an Afex treatment mostly to break down the lignin at this point. But that is a good question. We don't pressurize them at that point.
Great, thanks.
Sure. Secrets. Shh, don't tell. Okay. So we have our microbial fermentation to biofuels, but the research that I do is actually kind of at an even further downstream step in the pipeline. We take this stuff called conversion residue, which if you read the papers and stuff for today, that's also previously called stillage because it's what's left in the still after they remove the biofuels. And then subject that to another round of microbial fermentation to get useful bioproducts. And this isn't really a very new idea. We got this idea from the petroleum industry because one of the ways that petroleum is so cost effective is they use every carbon out of the barrel of crude oil that they get. A lot of it goes to gasoline, some of it goes to jet fuel and other products, but they use all of it. And so people at the center kind of got to thinking, well, if we used more of our plant matter for products instead of just what they do now with the plant matter that's left over is they burn it. And then the energy they get from burning it, they use to power the plant, and anything extra they sell back to the grid. So they're like, well, if we make something else out of it, could that get us more money than just burning it? So there are a lot of challenges to this kind of project, and I just want to point those out real quick. Doing this sort of thing, you're working at the end of the pipeline, which means if anything upstream changes, then the sugars that you're left over with in this conversion residue for your microbes to make into bioproducts might change. If they use a different microbe to make a biofuel, if they make a different biofuel, that microbe might have different waste products that your microbe would have to deal with in the conversion residue, or there might just be different sugars overall. And so you kind of have this whole, what I'm working with might change at any time. I think I see a question. No, you'll interrupt me. It's fine. I have been told.
Sorry, I'm including links to the readings for those
Oh
who
yeah, go ahead.
for the class.
It's in the more box. It's hard to see. So the second challenge that I ran into when I started with this project is there are shifting economics and shifting markets. So when we talk about making a valuable bioproduct, what does that mean? Well, today that could mean that you might want to make a lot of, say, limonene or isoprene, but tomorrow you might want to make a lot of something else like fatty acids or some other molecule. So when you put the two of these together, you kind of have this whole issue of, I don't know what I'm going to be starting with, and I don't know what I'm going to want to make, but whatever it is, I had better be able to make some money doing it, and I'm going to have to engineer some microbes to do it. Okay, no problem. So where do you even start when you're given something big like this? Well, I decided to start with what microbe am I going to use? Because I'm going to have to engineer a microbe. Which microbe do I want it to be? And the answer that I came up with was this microbe called Streptomyces. So Streptomyces is a soil microbe, and it undergoes this unique life cycle where it starts off as a spore, and that kind of germinates and grows these vegetative mycelia and hyphae, almost kind of like a fungal growth. And then it makes spores again, and then it can live as a spore for a long time. Now, the reason that I picked Streptomyces is because as a soil bug, it's going to be around a lot of different other microbes, so it should be able to handle different waste products. And it also eats a bunch of different material that you would find in the soil, like decaying plant matter. And so if it lives in an environment where it might come into contact with the sorts of things I want it to eat, and it can grow on a wide range of these carbon sources, then it might be a good bacteria to use in terms of flexibility. Because if we don't know what we're making and we don't know what we want, we really just need to make a very flexible system that can accommodate a whole bunch of different changes. So Streptomyces, not only can it grow on a wide range of carbon sources, it's been used industrially to produce what's called secondary metabolites. And those are things like antibiotics and insulin and all sorts of other products that you can use a bacteria to make that it doesn't need for its own growth. So if you have it making something else, that's good. So it can grow on things that would be in conversion residue. It can make the sorts of things that might become valuable bioproducts. This sounds great. We're addressing all of the concerns. What about the engineering? Well, there are some well-developed genetic systems that you can easily transfer between different strains and species of Streptomyces so that you can do genetic engineering. And so a lot of my talk from here on out is going to focus on this genetic engineering that I've done for this organism and the why and the how of that. So I just want to introduce you to the genome a little bit and the genetics of Streptomyces.
So we might
Yeah,
interrupt. We got a couple of questions
please.
in the Q&A. So first one was, what are you and your research center doing to avoid corn ethanol problems, such as the end-to-end, more energy is consumed than produced, and then also, carbon emissions? They're not the carbon emissions of corn ethanol aren't necessarily all that much better than fossil fuels. So that's one question: is sort of the energy in versus energy out, and then carbon emissions.
Well, we have switched away from corn. No more corn. All done with corn. All of the work that I'll be showing you today is done on conversion residue generated from switchgrass. And I know a lot of people are talking about switching from switchgrass to poplar and some other different things, but there's no more plans to work with corn anymore. And they're also moving away from ethanol as well, although a lot of the conversion residue that I have here was generated using a bioethanol distillation. I think they're talking about now moving to isobutanol.
Right, and I might add, so regarding sort of the energy inputs, I think the idea is to rely on perennial grasses that don't require as much input for planting and for fertilizer and pesticides and things like that. So that reduces the energy that goes into it. And then for the carbon emissions, if you're using perennial, then much more of the carbon is sequestered in roots in the soil compared to corn. And then a second question that we had is, at what scale is this process economically viable?
Oh, you have hit on one of the most important and valuable questions that we have been working on for many, many months. And I can talk about that now, or I can talk about that at the end. It's entirely your call. But that is a big question. Preference?
Let's go through more and then maybe address it at the end.
Okay. Don't let me forget. Save that one for the end. Very important. Because even the higher ups definitely ask that all the time. All right, back into the nitty-gritty of the Streptomyces genome. So most bacteria have a circular genome, but you'll note with this diagram of Streptomyces, there's this open area up here where the two lines of the genome do not connect. And that's because Streptomyces is unique in that it has a linear genome. And so instead of it connecting and making a circle, it's a line. And if you look, there's this kind of dark blue and light blue ring around the outside. And what that indicates is the kind of two parts of the Streptomyces genome. There's this center part here in the dark blue, which is what we call the core of the genome, which has the central metabolism, primary metabolites, any of those things I was talking about that the cell needs to maintain its own functions. And so that's the stuff the cell needs to be able to survive and grow and eat and live and reproduce. And then down at the end, there's what we call these are the arms. And these are kind of the more flexible areas of the genome where it produces secondary metabolites. And that's the area of the genome where we do our genetic engineering. And that's also the area of the genome that the cell will kind of change around to mutate and to be able to produce new products. And that's kind of the changing variable region of the genomes. So it's kind of neat if you think about this organism. It only needs this to live, but it spends so much time and energy maintaining all of these other processes here to give it competitive advantages in its natural environment. So when I talk about working with Streptomyces, I also just want to say I'm not working with one specific strain of Streptomyces. I actually have a collection of 85 different genetically diverse Streptomyces, and this is a phylogenetic tree or like a relatedness family tree for Streptomyces. And each of these little branches here is a different strain that I have to work with. And that's actually a really useful tool because even though a lot of the kind of central metabolism stuff might be the same, those secondary metabolite arms of each of these strains are going to be very different. And that's going to give me a lot of kind of genetic material to work with when I'm trying to keep these strains flexible and change what they can and can't make. And that's kind of a really great advantage from the engineering point of view. So now that I've introduced the organism that I want to use, what are we going to make? What are we going to do with these Streptomyces? And the thing that we decided to go after were these things called isoprenoids. So isoprenoids are a class of molecules that actually encompass a lot of different things. And I'll show more of what those are later, but they encompass stuff that we use for food production, makeup production. There are some fuels, chemicals, and a lot of different compounds that can be very valuable. So what is our plan for tackling this? First step of the plan, construct a reporter. And that's just a genetic element that will kind of say, can these strains make isoprenoids? Once we know if they can make isoprenoids, how much? How much can these guys make? We've got 85 different strains. Are some of them better at making isoprenoids? Because I want to work with one of the ones that's better at making isoprenoids. And then once I find a good producer, how can I have it make more of these things? And then once I know that I can make more of these reporter molecule that I decide to make, how can I have it make other things that are more valuable? Seems straightforward enough. A simple 4-part plan. Let's see how it goes, shall we? All right, so isoprenoids. I'm going to show you now a metabolic map of the cell. And this is just kind of a blueprint for how the cell is going to construct the molecule that we want it to make for us. And it's going to take glucose from its environment, from the conversion residue or whatever sugars it can eat that are left over. Probably there won't be any glucose left. But normally isoprenoids go through then glycolysis, which is kind of the main central pathway for breaking down sugars in the cell that makes these two molecules, glyceraldehyde 3-phosphate and pyruvate. And then our carbons are going to flow through this MEP pathway. Until it makes these two molecules, DMAP and IPP. And you'll notice that there's this kind of blue and red area going right there where there are little 5 carbons that are highlighted in blue. And that's kind of how isoprenoids are going to work. So DMAP can immediately be converted into isoprene, which is a fuel molecule and a valuable molecule that is of use that we'll talk about a little bit later. But if you take DMAP and you add on another 5 carbons, you get this kind of 10 carbon GPP molecule. And then you can take that, and once you have this carbon backbone, you can use a bunch of different enzymes called isomerases or synthases, and it can actually construct any of those different molecules from that 10 carbon backbone, which is really neat because you can then take that one molecule, make a bunch of different molecules from it fairly easily. And this kind of isoprenoid synthesis pathway keeps going. You can add another 5 carbons and get a 15 carbon backbone that you can make into a bunch of different molecules. And you can keep going this way on and on until you start getting bigger molecules like 20 chains. and these 15 chain molecules, and those you can combine like two 15 chains to get a 30, two 20s to get a 40. And so you can get all the way from this 5 carbon molecule to something with 40 carbons just in this one pathway, with all of these potential molecules that you can make just from the central pathway branching out kind of at these side steps. So looking at this, we decided that the best thing to use as a reporter is this molecule way down here at the end called lycopene. So just like that box, lycopene is a bright red color, and you guys are probably familiar with it as the molecule that makes tomatoes red. So you all are familiar with lycopene and its brilliant redness. And this seemed to be a good reporter molecule because it's at the end of the pathway, which means that if you can make lycopene, you can probably make anything else along this pathway, and it turns red. So if the cells are making it, we will know, and we can look at how red they are and get an idea of how much potential they have for making these isoprenoids. So how do we turn cells red? Well, it's kind of a fun thing. And I just want to go into then a little bit about genetic engineering. So I'm going to talk a lot up ahead about genes and gene expression, and that's just how much the gene is on, how much of the molecule are we trying to make. And I'm going to talk about promoters, which I'm showing in this little here molecule, this little arrow. And that's just the thing at the start of a gene that lets you know that it's being made. So you can actually control how much of this product you make based on controlling this promoter up here. So how does engineering a microbe work? Well, with E. coli, life is pretty simple. You have a bacteria. It has what's called a plasmid in it, which is this little free-floating bit of DNA. You take the plasmid out. You put the genes that you want into the plasmid to have it do whatever you want it to do. You put it back in your bacteria, and all of a sudden, it makes whatever that foreign DNA tells it to make, whether it's antibiotics, insulin, enzymes to break down cellulose, ethanol resistance, whatever change you want to make to your cell. And sometimes it'll mean you only need to add one gene, sometimes you'll need to add a couple, but pretty straightforward. Now, Streptomyces lives in an environment with a lot of different organisms, and it's really not in its best interest for any other DNA to be coming in there willy-nilly and messing with it. So if you want to get DNA into Streptomyces, you have to first do this with E. coli, and then when you have this E. coli with this plasmid in it that does what you want, you use that E. coli to transfer that plasmid into your Streptomyces. So your E. coli kind of acts as a middleman for taking that plasmid and putting it into your Streptomyces. Now, Streptomyces also cannot maintain a plasmid, so it can't have this separate thing here. So it then takes that DNA that the E. coli puts in it, and it puts it right onto the chromosome. So that means that in order for your cell to live, it's going to have that little bit of DNA in there. Now, from an engineering point of view, that means when we're making this plasmid, we can't just put in the genes that we want. We also have to put in what's called the transfer machinery. That's the machinery that gets this plasmid from your E. coli into your Streptomyces. Chromosome integration machinery, which is a fancy way of saying the stuff that puts it on the chromosome there. And that includes a recognition sequence, so where in the chromosome is it going to put it? And integrase is the enzyme that puts it in at that spot. So it's kind of a neat little system because you're like jury rigging this thing, like, okay, we got to do this and that, get it in there. So how did we decide to design our reporter? Well, the first thing we really wanted to focus on was, like I was talking about before, these promoters. So if we put in a promoter that's too strong, it's going to make so much of our molecule that it might be toxic to the cell. And if we put in a promoter that's too weak, it might not make very much of it. And so when we do our assays, we might not see that it can make any of the molecule that we want. But how much is too strong and too weak is very dependent on the cell itself. And that's one of those things that when you start off, you maybe don't know how it's going to work. So luckily, this previous researcher had come up with all of these different promoters, and they're called synthetic promoters because they're made so that they're always on. They're constantly going to be making whatever you want, but at different strengths. And you can see here, this is just a little graph of how strong these promoters are. And we decided to take one that was really strong, one that was kind of in the middle, and one that was a little bit weaker, just so we'd have a range of different things to test how our cells would behave. And we combined these with ribosome binding sites, similarly strong, weak, and kind of middle. And we constructed that plasmid that I was talking about, where there's this integration machinery and these 3 genes that we need to make lycopene, and this transfer machinery to get it from E. coli to Streptomyces. And when I did this, I was actually only able to get the weakest promoter that we picked to work with the E. coli. When I tried putting any of the plasmids with the stronger promoters, the E. coli just did not grow at all, just dead. Very, very sad. But that's okay. I was able to get one to work. And so then I took the one plasmid that I had that was designed to make lycopene. I put it in my E. coli. I conjugated it into all 70 some of those different streptomyces that I had. And then I looked to see if they turned red. And obviously, I probably would not be so excited about talking about this if they hadn't, so they did. And you can see here, these are just 3 different strains from that collection that I was talking about of all those different streptomyces. And the numbers of the strains are just their ID numbers here on the plate. But I took the original strain, a strain that had that plasmid without my lycopene genes, kind of as a control, just to make sure adding a plasmid isn't going to mess anything up. And then the plasmid with those 3 lycopene genes. And you can see that these different strains actually had very different amounts of native lycopene production. So when I went and mapped this onto my collection, so this is again that map that I showed you before that was a big circle. I'm just stretching it out here so you can see all the different names of the strains. And I looked at the strains that were able to grow well on conversion residue. And this subset down here were able to produce a lot of lycopene. And that's probably not very surprising that the strains that were able to make the most lycopene are also very closely related, because they probably share a lot of those same genetic elements that make them good producers of lycopene, and hopefully of isoprenoids in general. So now that I have strains making lycopene, yay, how do I get them to make more lycopene? Well, the answer to this actually lies upstream. So the thought here is if we want them to make more lycopene, we also want them to make more of all of these other things here that go into making lycopene. And one of the places where there might be a bottleneck in this pathway that we could try to overcome is up here with the MEP pathway. So the MEP pathway is going to take carbons away from central metabolism, which is the pathway that you need to make all those things you need to live as a cell, and kind of shuttle them through this isoprene synthesis pathway. And so how can we use the MEP pathway to get more carbon to lycopene? Well, the Smansky group up at the University of Michigan actually was able to come up with the way of doing this using those same promoters that I was talking about before. And they took all of these different synthetic promoters and they changed the promoters for each of the genes in this MEP pathway. And they took every possible promoter and gene combination and tried them pairwise, 3 at a time, 4 at a time, 5 at a time, until they had built an optimized MEP pathway using these synthetic promoters that would pull as much carbon as possible away from this central metabolism down to the isoprenes without killing the cell. Very important, without killing the cell. And so when they did this, I took their pathway and I put it in my cells. And I did the same thing that I showed you before, where I put it on that plasmid and conjugated it into the cells. And you can see from the data here, if you just have the empty vector, not very much lycopene. If you have just the MEP pathway, not very much lycopene. If you put in the lycopene reporter, there's a bunch of lycopene. But if you put in the lycopene reporter with this optimized precursor pathway so that you get more carbon toward there, you get more lycopene. Just super great and exciting. This is exactly what we had hoped to see and was really very encouraging. So there is another way to work at increasing the precursors. I'm not going to talk about it very much, but I just want to mention there is another pathway that can get you kind of to the isoprenoids as well. And the MEP pathway is what is used in streptomyces, but the MEV pathway is found in E. coli and other organisms. And so the thought being, if we took the pathway from other organisms and put that in here, could we get that sort of same result? And I've just started working with it. And the answer so far appears to be yes, but I don't have any data comparing the two different pathways to show you right now, other than it's very encouraging results as well. Okay, so now that we can make lycopene, what's next? Well, the next step is to make other more valuable things. And so I wanted to start kind of higher up in the pathway with isoprene, which is a valuable fuel molecule, pinene, which is a chemical compound that's often added to cleaning reagents, and limonene, which is often a food additive. And I'm sure you can guess by the names, pinene normally comes from pine trees and is that piney smell, and limonene normally comes from citrus crops. And so a lot of these can have very variable markets depending upon the plants that they come from. So in order to do this, I had to do more of this engineering and more of this making of plasmids and making of pathways. So looking at the MEP pathway, if I want to make isoprene, I'm going to need to introduce an isoprene synthase to get that from DMAP to isoprene. And if I want to make pinene or limonene, I'm going to need the pinene or limonene synthase, but I'm also going to need a GPP synthase, an enzyme to do this next step in the pathway that converts it from 5 carbons to 10 carbons. And this is very important because the enzyme that is normally here actually is bifunctional. So it does this GPP synthase activity and it also does FPP synthase activity. So if you just use the enzyme in the cell, it's going to convert DMAP possibly directly to FPP, which is the 15 carbon molecule without stopping at the 10 carbon molecule like you want it to. This GPP synthase is a single function enzyme, and so it'll just catalyze that first step. So you'll have an increase, hopefully, of 10 carbon chains that you need to make your pinene and your limonene. So luckily, I can just use the same backbone that I used for limonene or for lycopene, because I'm just going to be replacing the genes that make lycopene with the genes that make isoprene, or the genes that make pinene or limonene. And so that part's pretty easy, but you actually have to make a whole bunch of different constructs. So one of the difficulties in engineering like this is that the genes that we're using come from different organisms. So like I was talking about, they come from white fir, poplar plants. I think this guy right here is a sweet potato plant. And just because the gene works in a plant doesn't mean it's going to work when you put it in a E. coli or a streptomyces. So you have to do a lot of engineering of the gene itself to try to optimize it to work in your organism. So when doing this, we actually took genes from a bunch of different plants to see if one or the other is going to work better in our organism. So we have here 4 different isoprene synthases that we took. And these ones were a collaborator of ours optimized these for streptomyces. And so what that means is he went through the genetic code for each of these genes and tried his best to make sure that it would work in streptomyces. And the sorts of changes that you have to make are things like GC content and codon usage and regulatory element avoiding. It's actually it's actually in some ways kind of easy because nowadays they have computer programs that can do this. You just put in your genetic sequence and say, please make this work in E. coli, or please make this work in streptomyces, and it spits something out. But it's also somewhat labor intense because when it spits something out, that doesn't necessarily mean you can just make that. It says, this is what you should do. And then you're like, but you can't do that sometimes. And so you really have to work with that sequence and try to do a lot of optimizing, figuring out this might be what would be best in streptomyces, but if I go to a company and say, can you make me this DNA? Can you engineer this sequence for me? They might say, no, if we try to put that many C's in a row, our machine will just break and we can't do that. So you really have to look at how to kind of mesh what is technologically possible versus what is theoretically optimal, which is why we do this sort of thing. And then from there, we also engineered 2 versions of a limonene synthase plasmid using 2 different GPP synthases. and to pinene synthases, again with these same different GPP synthases. And so this first GPP synthase was found in a plant, Habeas grandis, and is one that has been used a lot commercially. But when I was going through some sequences, I found that there's actually a Streptomyces it's not very well known. It's a lesser used strain, but it has this single GPP synthase instead of a bifunctional one. It's kind of unique. It's also one of the few Streptomyces that has that MEV pathway in it. So it's kind of a unique bug that I've been playing with in terms of its relevancy for this sort of projects. And then just up there is the papers where I got those guys from. So right now I have put these plasmids into my Streptomyces, and you can tell by the way they smell, which is, I know, a horrible, awful way to be testing organisms in a laboratory environment, and it's definitely made more challenging by the wearing of masks. But you can smell kind of this lemony smell, and isoprene synthase, it smells very jet fuel-y. And right now we're working with the core facilities at the GLBRC to try to get detection on these molecules and get an idea of how much we're actually producing, because eventually if you want to use these industrially, you want to know how much you're making. So the goal of the plan was to produce isoprenoids from conversion residue. And so we engineered a lycopene construct as a reporter for the production of isoprenoids, determined which strains had the most of this lycopene production. We introduced a precursor MEP pathway to increase production, and then started producing other isoprenoids that could be more valuable molecules. And the way that those plasmids were made, you can actually just kind of almost cut and paste which isoprenoid you make whenever you want to make one. And so the genetic engineering from there becomes a lot easier because it's kind of like this hot swap system for the genes you might want to make. And then this fits all back into that whole big pathway that I was talking about with the GLBRC earlier. Because now that we have this kind of flexible organism that can eat a whole bunch of different plant matter and make a bunch of different bioproducts, that can kind of work hand in hand with a lot of the other upstream elements. And we can start using this maybe up here when we have this initial breakdown product. We can filter some of the stuff off that we know can't be metabolized into biofuels, and that can go straight into making these bioproducts. And it kind of just brings everything all together as another way just to get rid of some of the waste of the system and try to make something more valuable out of it. And with that, I have to acknowledge these so many people. Don't worry, I won't talk about all of them. The rest of my lab is always helpful. The Curry lab are the ones who gave me a lot of the initial strains to work with the Streptomyces. These are some of my collaborators working with how the bacteria use conversion residue. The Amador Nuguez lab has been helping me look at intercellular metabolites so I can watch and see if carbon is being pushed through the pathway how I want. Core facilities makes us conversion residue and studies the metabolites our strains are using. Allison actually put together the first part of the talk about the GLBRC. She's their outreach coordinator. She is amazing. And then I have other collaborators all over the GLBRC and outside. Christos does the economic analysis with Shamik, which I'm told I will be talking about next. And then Trey heads up a lot of the biotech stuff over at the core facilities as well. And that was what I had prepared for you guys today. Please feel free to look at more stuff. This is from the last time we all met in person at our annual science meeting, and all of the members of the GLBRC try to get together and talk about their science and coordinate and learn new things. So if anyone has any other questions first, then I'll talk about that. Otherwise, I'm going straight into the economics.
Looks like we have a couple of other questions that came in. One from Raj is, does GMO plants show any difference?
They do. A lot of the modified plants that have been engineered specifically by GLBRC for GLBRC purposes have lignin, which breaks down more easily, and so, or less percent lignin total, so less of that goes to waste, and they're meant to be broken down in that way. And other ones they're trying to work on engineering more of the cellulose portion and less hemicellulose, just to get more 6 carbon molecules instead of 5 carbon molecules as a food source, because the more carbons you start with eating, the more you can make with them. Is that what you meant? They're also working on their ability to nitrogen fix and use the land better so that they work on marginal lands. I know it's another engineering aspect of it.
So I guess if there's a follow-up, I'll let you know. Another question, do you consider examining a pathway for obtaining effective microorganisms?
A pathway for effective microorganisms in terms of?
I'm not exactly sure.
You can look for pathways that you think would be useful in your system. And then if your organism doesn't have one, you can try to engineer it in. And that's something we definitely are doing. And that's why we looked at so many different strains of streptomyces, because we're like, well, what if this strain is really great at producing isoprenoids, but it just can't use xylose for some reason? And there's a lot of xylose in conversion residue. How do we, and then, if we can take one of the streptomyces that can use that xylose, then we can take that pathway from it and move it in where we want it. So a lot of pathway engineering and moving has been utilized. And we also have people at the GLBRC who look for novel pathways, pathways to use things that no one's thought of using before, and analyze how does it do that. And then we think about, well, is that something we'd want to put the two bacteria together and let them work together? Or is that something we'd want to take the pathway out of the one and put it in the other? And a lot of that depends on how easy it is to do the genetics of the organisms you're working with.
Then a question that came in the chat, could the MEP pathway method be applied to current crop residues like corn stover and soybean stubble?
The optimizing it pathway?
Not sure. I think, and yes, it's the follow-up.
Yes. So the pathway optimization can be used for any pathway that you want to engineer. That specific one was designed using promoters specifically for streptomyces, and it was totally made and designed and tested in streptomyces. A lot of people talk about pathway optimization, and when they do it in practice, they'll do a metabolite analysis to kind of see where the bottlenecks are, and then they'll just engineer the one or two enzymes that are shown to be stopping the pathway, and then hope that fixes it. Because if you can just engineer one enzyme a little bit and it fixes the whole thing, that's a lot less effort than, it's a supplemental slide, and there's always this pressure to bring up a supplemental slide when there's a relevant question. So I'm gonna do it. But when you do the pathway optimization, that pathway was, where's it? Yeah, was 6 genes, 7 genes. And you can see in this map, you see all those crossing lines. That's every single comparison that they tried. So the optimization itself and then testing every single resulting thing took months and months of work. And if you can just engineer one enzyme, it's usually a lot faster. The reason that they did it this way for the MEP pathway is different reports from different organisms had different bottlenecks that they had shown. And so they weren't sure whether the bottlenecks were very specific to the organism you were testing. So in order to get around trying to figure out if the bottlenecks were specific to the organism, they did this all in vitro in the test tube, so that then they could just say, we put the carbon in, we tested the carbon that came out, this is the best one. We don't know what else in the cell might be messing with it, but if you just look at the pathway all by itself, this is what you get.
A couple of questions related to efficiency. One was, what is the conversion efficiency of the pathways you've studied? And another is, does reaction time for different pathways indicator of efficiency?
So efficiency in this case ties into the economics of it, because the longer you have it growing, the longer it's going to take to do things. And a lot of it comes down to the turnover. Now, with a cell, with a living system, the efficiency that you can get out of the cell is also related to how well the cell grows. You don't want the cell growing too much, because then it's spending all its energy growing and not making the product you want. So, but if you make too much product, then the cell will die and you don't have any cell to make the product you want. So there's this really delicate balancing act of how do I get as much as I can get without destroying my production plant, for lack of a better term. And so the efficiency that we get in terms of conversion residue. There's like 60% of the biomass left over after hydrolysis. Our bugs can then eat another 40 to 60% of the mass, and then we convert that to 0.5 mg per mil lycopene. I mean, there's a lot of loss, and it's not converting all of it, certainly. But is it better than burning it? Yes, you could probably sell it for more than burning it. Are we ever economically going to replace petroleum? Currently, no. Petroleum is super cheap. You can make a lot of things with petroleum. A lot of this is just going into the whole, can we make this process sustainable? Can we make enough money doing it that it's cost neutral? And as the environment changes, as the economics change, as the policies change, these are the technologies that they're going to be reaching for, and we need to have them ready. So a lot of what I do is it better than burning it? Yes, yes, it is more economical than burning it. Are we going to totally make biofuels cheaper than petroleum by doing this? We don't know. I mean, at some point, it could turn out that one of these isoprenoid bioproducts, like a C25 cancer drug, is really, really important to make. But it's really impossible to know at this point how it's going to turn out and when. And that's one of the economic issues that we've definitely been struggling with is if we can't say it's cheaper than petroleum, how do we justify it? And a lot of that is just future sustainability.
And that sort of gets back to the original question about economic viability.
It's a very thorny problem. That's why we have economists.
Another question that came in, they're wondering what is done to prevent genetically modified organisms or their engineered genetic features from escaping into the wild or taking root in the wild.
Well, we have to sign a lot of paperwork saying that we're going to be disposing of them properly and handling them properly. They're not allowed to go down the drain. In fact, we're not even allowed to use ethanol to kill them because ethanol won't kill spores. Any genetic material and any live cells that we use immediately get put in an autoclave, which is a giant pressure oven. If you haven't ever worked with one, it gets up to 120 some degrees and 20 some PSI. And then we prove that they're absolutely totally dead. And then we are allowed to put them down the drain and dispose of them and things like that. There's also a whole bunch of antibiotics we are not allowed to use, not allowed to genetically engineer our bacteria to be resistant to, because that is a concern that has always been present. If the more we work with those antimicrobial resistance genes, the more they could find their way into the wild. And so for things like, was it Viomycin? I think it's okay. Is it vancomycin? There are some like drugs of last resort antibiotic wise that we are not allowed, no one in the country is allowed to engineer their bugs to be resistant to those sorts of things. And other than that, a lot of the genes that I'm using actually came from other streptomyces. So I know I'm working with those plant ones, those couple plant ones that are new, but before that, they were all things that were already in the wild. And a lot of streptomyces even have the genes to make lycopene. They're just under the control of promoters that only get turned on in very bizarre circumstances. I believe with lycopene, it's blue light. So if you expose the streptomyces to blue light, then they produce lycopene. I can theorize about, well, I can tell you what the paper theorized about why that happens to be, but a lot of that material is kind of already out there. And even with plants, I mean, those plants are already out there. So we're not making anything totally foreign.
Another question that just came in, how does GLBRC decide what products to target? Is there a pathway to viability in mind, a government direction? It makes sense that you chose isoprenes, but I'm curious what else went into the decision and how researchers choose these products in general.
So Christos from my acknowledgement slide is the economic guru. Shamik is the postdoc that works with him who's been collaborating on my project. And they do a whole bunch of techno-economic analyses. And so they ask you questions like, how many microliters of NaOH did you use to pH that? And they really get down to the nitty-gritty detail of everything that goes into the process to kind of try to calculate with, okay, if this is everything that went into the process, this is what it costs to purify it, this is how much we can sell it for on the open market, and they're doing the economics for us for some of those molecules right now. They, a few years ago, did one with limonene, which is kind of why we targeted it. And pinene was one that we had read was, it generally comes in an alpha and beta form. And so if we can gear our organism to making one of them, which is more economically viable, or we can separate it out, because one of the problems in that pathway is just separating out the two forms of pinene to use the valuable one. And so some of the molecules like that are things where it's like, well, this one just, we've been told might have a market. And with limonene, it's very valuable. If you have a good citrus crop, limonene is not worth very much. If you have a poor citrus crop, limonene can be worth a lot. So there's one of those, well, let's target it. And a little bit from my point of view was I kind of wanted something I could smell and know it was working, because that was just a way for me to test it. Like I could see the lycopene was red, and we thought starting with 5 or 10 carbon molecules, that might give us an idea of kind of the maximum production we could get. So lycopene is kind of tough in that it goes in the membrane of the cells. So you can only make as much lycopene as the cell can hold kind of before its membrane falls apart. With isoprene, limonene, pinene, those are all excreted and they're smaller molecules. So the thought was we might be able to get a higher yield off those and then get more of an idea of our full range of production potential. So there was a little kind of economics said these ones are good, and then just, well, these are also experimentally informative, hopefully.
Great. And then another question that just came in, what are your thoughts on production of ethanol from Zymomonas mobilis?
I am really glad that the people are currently producing bioethanol from Zymo. That's the conversion residue that I get and work with most of the time. We are working on a synthetic conversion residue, which has been a really interesting process. I think they're still going to use Zymo, but I do think they're going to isobutanol.
Great. Give one last opportunity for questions. Otherwise, we'll wrap things up here in a minute or so. Looks like we've got, so are there any life cycle assessments of biofuel production from switchgrass to support the carbon emission reduction goal?
Wow, I have no clue.
There definitely are. I know there have been researchers involved with GLBRC in the past who have done like life cycle assessment of biofuels. There's another, I think it's Argonne National Labs has a program called the GREET model, GREET, that you can assess different emissions from different transportation fuel sources. I believe that biofuels like switchgrass are involved in that. So I do know that there are assessments out there. I'm not sure with the current GLBR research what's out there. All right, well, thank you so much, Dr. Wadler, for your presentation today, and thank you all for your questions. So next, we'd normally be having a presentation 2 weeks from now, but instead we'll be switching over to, there's an event that the Wisconsin Energy Institute is hosting on April 7th called Solve Climate by 2030. It is a panel discussion talking about statewide solutions for Wisconsin in addressing climate change in the near term. If you're interested, I'll type in my email address here. Actually, I can link in the chat as well. And so that'll be on April 7th from 7 to 8:15. One of our featured panelists is actually Lieutenant Governor Mandela Barnes. So we hope to see as many of you attend that event as possible and listen in on the discussion. So thanks all for joining us, and we'll hope to see you on April 7th.